I want to talk about a special case of diffusion. This special case of diffusion is going to be called osmosis. Osmosis is the diffusion of water across a membrane. This is very important to our cells, and it helps our cells to maintain homeostasis on a cellular level. When we talk about the movement of water across a membrane, we can talk about the concentration of water across a membrane. So typically when you look at a solution, you can either look at the concentration of the solutes or the concentration of the water. It doesn't matter which one you focus on, you just need to figure out which one makes the most sense to you and stick with it. If you have a low concentration of water in a solution, that means you have a high concentration of solutes. The opposite is also true. If you have a high concentration of water, you have a low concentration of solutes. So you need to think about which one makes most sense to you when you see a picture of a solution. And that's the one that you want to stick with while you think about osmosis. Now, in order for osmosis to occur, our membrane must be freely permeable to water. And in general, when we talk about osmosis, it is our membrane will be selectively permeable to solutes. So from now on, when you think about a membrane, I want you to think water can move across no problem. Always allowed to move across. But our solutes are going to be stuck on one side or the other. These solutes are not going to move. Okay? So when you think about membranes, water can move freely. Solutes cannot move freely. They need help. They need a carrier protein or a channel protein or something like that. So, in the process of osmosis, water molecules are going to move down their concentration gradient, meaning water molecules move from an area of high water concentration to an area of low water concentration. If you are looking at this from the perspective of the solute, that means that water is moving from an area of low solute concentration to an area of high solute concentration. Now again, it doesn't matter which way you look at it, you just need to pick one and stick with it. The way that I like to think about it is I like to think about the solutes. So it's a little bit backwards, but I think about the water diluting the solutes, making the concentration of solutes equal across the membrane. In order to do that, we need to spread out that high solute concentration and make it more dilute. That's generally the approach that I use, but you need to use whatever works for you. Alright, when we talk about osmosis, we can talk about osmotic pressure. Osmotic pressure is the force of the water's concentration gradient. The opposite of osmotic pressure, the force of the water, is called hydrostatic pressure. Hydrostatic pressure is the force needed to block osmosis. And so osmosis will continue until our osmotic and our hydrostatic pressures are equal. When we talk about osmosis, we can also talk about osmolarity. Osmolarity is the total number of solutes in a solution. This can incorporate different kinds of solutes. So if you put table salt into water and you also put sugar into water and you mix it up, you are going to have a solution that has glucose molecules as solute number 1, sodium ions as solute number 2, and chloride ions as solute number 3. So osmolarity is the total number of solute particles in a solution, but it includes many different kinds of solutes. Tonicity, on the other hand, is going to refer to the solutes that have the ability to change the shape or the tone of the cells by altering the internal water volume of a cell. So tonicity is going to have an osmotic effect on a cell. When we talk about tonicity, we are talking about solutes that are not capable of moving across the membrane. Because these solutes are not capable of moving across the membrane, water is going to be the only thing that can move across the membrane to alter the concentration on each side of the membrane until we get an equal concentration of water molecules to solute molecules. Now, the interesting thing about osmolarity and tonicity is that two fluids may have equal osmolarity. They can have the same number of solutes in their solutions, but they can have different tonicity based on which solutes are present on each side of the membrane, because tonicity refers only to solutes that will not move across the membrane, whereas osmolarity includes everything, including some solutes that can move across the membrane. We are going to focus on tonicity and the ability to alter the internal water volume of a cell. We're going to have 3 different categories of tonicity. A solution can either be isotonic, hypotonic, or hypertonic to something else. The important thing to remember about these 3 terms is that they are comparative terms. So we can talk about the cell is blank to the beaker, or we can talk about the beaker being blank to the cell. And depending on which of those sentences we use, the blank, the vocabulary word that goes in the middle, is going to be different. Okay, so let's start with isotonic. An isotonic solution is a solution that does not cause water to flow in or out of the cell. This means we have equal concentrations of solute on either side of the membrane. So our cell is not gaining water and our cell is not losing water, because we have equal concentrations of solute on either side of the membrane. In a hypotonic situation, hypo, that prefix, means below or under. Tonic is referring to the solute. So hypotonic means that we have less solute than the other side of the membrane. This means the other side of the membrane has more water than whatever we are discussing. So let's use the sentence, the cell is blank to the beaker. If our cell is hypotonic to our beaker, our cell has less solute than the solution the cell is sitting in. If our cell has fewer solute molecules, then our cell is going to lose water, because the water is going to be in high concentration inside of the cell and low concentration outside of the cell. So water moves down its concentration gradient. If you're the kind of person that takes a look at the solute, your cell has less solute, and water is going to move towards the area of higher solute concentration. So our water leaves the cell to dilute the solution outside of the cell. Our cell loses water, and our cell shrinks. Okay? So if our cell is hypotonic to the beaker, our cell will lose water and shrink. This can cause the crenation of red blood cells when your red blood cells are hypotonic to the plasma. The flip side of hypotonic is hypertonic. Hyper means above, so hypertonic means that you have more solutes than the other side of the membrane. So if our cell is hypertonic to the solution, that means our cell has more solutes than the other side of the membrane. If our cell has more solutes than the other side of the membrane, it has less water. So if we look at it from the water perspective, our cell has little water. The outside of the cell has more water. So a hypertonic cell to the solution will take on water. Water moves down its concentration gradient from the outside of the cell to the inside of the cell. If we look at it from the perspective of solutes, then our hypertonic cell has more solutes than the outside of the cell. Water always moves towards the area of higher solute concentration. So in order to dilute our high solute concentration, water moves from the outside of the cell to the inside of the cell. This is going to cause our cell to swell. This is dangerous because our cells can swell to bursting and actually pop open. And if we rupture a cell like this, that is going to kill the cell. When this happens to red blood cells, it is called hemolysis. If we take a look at that word, the prefix hemo is in reference to blood, and lysis means to break. So we're breaking our red blood cell. This is dangerous. You don't want to do that. So if our cell is hypotonic to the solution, then the cell will lose water and shrink. That's crenation. If our cell is hypertonic to the solution, then our cell will gain water and rupture in hemolysis. Now let's flip our sentence around and let's take a look at the beaker is blank to the cell. Okay? If we do that, we're going to see different results. So if our beaker is hypotonic to the cell, the beaker has less solute, meaning the cell has more solute. So what direction is the water going to move in? If the beaker is hypotonic to the cell, water will move into the cell, and the cell will swell and possibly burst. Okay? So, as you can see, it's very important that you use these terms in a comparative sentence. You cannot simply say, it's hypotonic. First of all, what is it? Okay, you need to specify your subject. It is either the cell or it is the beaker, and it makes a big difference which one you're referring to. Okay, so specify your subject. The cell is hypotonic, or the beaker is hypotonic, and then you need to finish the sentence to the blank. Okay, because if your cell is hypotonic to the beaker, the cell will shrink. If the beaker is hypotonic to the cell will swell. Okay, so what happens to that cell is dependent on how you use your vocabulary. So please make sure to always use hypotonic and hypertonic in a complete sentence. Without the complete sentence, you don't know what is going to happen to that cell. Right? That is very, very important. That finishes our discussion on osmosis.